and gentlemen, good morning, good afternoon, and good evening. Welcome all of the LabClub members to join our first episode of Talk to Expert today. I am Shan, the International Hematology Marketing Manager of Mindray IVD. I'm honored to be the host of today. Before we get started, I would like to give you some background information about Talk to Expert. Talk to Expert is a series of online diagnostic panels exclusively on LabClub. We aim at providing an online platform for lab professionals to exchange their ideas with experts. This event will be held on every Thursday at the end of the month until November. So please stay tuned with our LabClub announcement for registration. Today is the very first episode. I'm very exciting. Let's take a look into the agenda. It will be divided into two parts. We are honored to have invited Dr. Khosrow Adeli today to talk about quality management in hematology. And then we will move into the second part and Q&A section. Dr. Adeli will be here to answer all of your questions. Please take this opportunity to ask as many questions as you can because we have got only 30 minutes. For now, please allow me to introduce Dr. Khosrow Adeli. Dr. Adeli is the professor in the Department of Laboratory Medicine and Pathobiology, Biochemistry, and Physiology at the University of Toronto in Canada. Also, he is the senior scientist in the Molecular Medicine program of the Research Institute. And he is the head of clinical biochemistry in the Department of Pediatric Laboratory Medicine at the Hospital for Sick Children in Canada. Furthermore, he is the president of the International Federation of Clinical Chemistry and Laboratory Medicine. Short form, we call it IFCC. I believe everyone knows this name, right? Now, please give a warm welcome to Dr. Adeli for his presentation. Dr. Adeli, please.
Thank you, Shan. Can you hear me okay?
Sure, yeah, it's okay. We all can hear you.
So I'm going to share my slides.
Yes. Okay, I will stop sharing. Yeah.
Well, thank you very much for this opportunity to present on quality management in hematology. Quality management in different areas of laboratory medicine, particularly biochemistry and hematology, are very similar. And I have been involved in quality management at the University of Toronto. We actually have a quality team, and that includes biochemistry, hematology, and other areas of laboratory medicine. So I'm happy to share with you my experience, which applies, as I said, to multiple areas of the lab. I'm at the University of Toronto, which is in Canada, specifically the Hospital for Sick Children. This is a large pediatric hospital in Toronto. I'll be happy, as Shan mentioned, at the end to answer your questions as much as I can. Now, I want to start by telling you about the importance of laboratory medicine. We have to remember that the lab plays a central role in healthcare delivery. As this graph shows, the lab is really playing a central role in identifying, when screening the population, identifying risk factors and symptoms, diagnosing disease, determining appropriate treatment for disease in many cases depends on laboratory testing. Also, evaluating response to treatment. For example, if you put someone on cholesterol-lowering medication, well, to evaluate response, you need to perform serial lab testing. So the lab plays multiple roles at different levels, and therefore really should be regarded as part of the multidisciplinary team at the center of healthcare. But to provide this important role, quality is critical, that the lab needs to provide quality results. Now, when quality results means having a very very comprehensive quality management system in place to ensure accurate and precise assays and test results. And that means having validated methods and systems. But in addition to that, interpretation and appropriate reference intervals are critical. That means one needs to not only provide accurate results, but also ensure that those results are appropriately interpreted. That usually means you need to have appropriate reference intervals or decision limits. Now, this is true in hematology, it's true in chemistry, but it's true in all areas of laboratory medicine. So therefore, my focus is going to be on quality management and reference intervals in hematology. But components of quality assurance, there are several components that one needs to keep in mind. To ensure a strict quality control management, one has to be monitoring quality at different levels, pre-analytical, analytical, and post-analytical. Pre-analytical meaning before you actually perform the test, ensuring that the sample is appropriately collected, the patient posture and patient collection is done appropriately, specimen collection and the equipment and the type of sample tube or vacutainer or syringe or whatever the equipment is, or the device for collection is appropriate to the testing. For example, in hematology, of course, one would like to collect EDTA whole blood samples, whereas in other areas, there might be other types of samples. So appropriate specimen collection is critical. Specimen transport and preparation and storage is really important, particularly in hematology, because samples, whole blood samples are pretty unstable, and therefore samples should be tested usually within 4 to 8 hours. In some exceptions, it could be refrigerated and tested a little later, but the sooner the sample is tested, the higher the quality of the results. Analytical steps are also important. That's really the actual instrumentation and the assays and ensuring that that's properly controlled. And I will discuss this in detail. We also have to be aware of post-analytical issues, how the data is collected and analyzed, and how the data is interpreted. That means appropriate reference intervals or decision limits. So what we call evidence-based clinical decision-making. That means you should not be using outdated reference intervals that may have been developed 30 years ago. You got to ensure that that data, the reference interval, is validated, it's verified, so that you have appropriate interpretation based on the latest technology that you are using. So that means you need up-to-date reference intervals for the instrument and the assays that you are testing. So next slide, I'm just listing a number of areas of hematology. Hematology lab services are, as you know, quite diverse. General hematology is something that everyone is familiar with, complete blood count, blood film, or bone marrow investigation are referred to as general hematology. There are specialized hematology areas for diagnosis of thalassemia, hemagglutinopathies, red cell enzyme defects, coagulation testing, of course, very common and an important part of hematology. Flow cytometry increasingly used. Here, for example, at our hospital, we have an extensive flow cytometry service to provide, basically to assist with diagnosis and monitoring of children with malignancies and immunological disorders. Molecular hematopathology is increasingly common. That means doing genetic testing to look at chromosomal translocations and look at basically childhood leukemias in our case, although this can also be used in adult settings for adult hospitals. Blood transfusion and stem cell biobank is also a service we provide at our hospital, and it's also important. Of course, at least in this hospital, is part of the hematology division. So the current presentations, today's presentation, will focus on general hematology, but you realize that it's much more elaborate. Hematology services can be a lot more comprehensive, but my focus today is on general hematology. So what are the key critical quality management aspects of general hematology? As I mentioned, pre-analytical considerations are really important. And actually, based on multiple studies done in Europe, in North America, in Asia, elsewhere, pre-analytical errors are most common. That means almost 70% of errors that occur in the lab or quality issues are related to before even the sample is analyzed. That means pre-analytical. So therefore, one needs to be aware of all of the factors that can cause these errors. So it's called root cause analysis. So you need to identify what the issues are and be able to address them. And you need to have steps to prevent these errors. Also, selection of the test is critical. So selecting the right test on the right patient at the right time is important. And so labs and lab professionals and scientists can actually assist clinicians, physicians, in selecting the right tests. So this is an important function that we provide, not only help provide the lab results, but also help the physician select the right test. Also, appropriate utilization is important. There is evidence of both overutilization, but in some cases underutilization. So appropriate utilization is critical. So here I have listed some of the common areas in the pre-analytical phase where errors can occur. Ensuring the appropriate test is requested, ensuring the appropriate patient ID is used. This is a very common error where the wrong patient ID is used, and that leads to, of course, incorrect test result and test reporting. Specimen collection, multiple issues can occur with that. Specimen transport and processing, as I mentioned, one needs to ensure rapid testing, usually, ideally, within a couple of hours, but up to 8 hours is acceptable. But this can be a problem, especially depending on the location of the lab and where samples are collected. This can be a challenge. So as I mentioned, a substantial body of evidence indicate that the major areas of improvement, actually, the focus of most quality control, quality management system should be on the pre-analytical phase because the pre-analytical phase is a major source of errors. Now, this table shows some of the common pre-analytical errors that you typically see, problems with the sample. Now, lipemia, for example, is a common problem. That means high lipid levels, lipids and lipoproteins. So when you have lipemia, lipemia is caused by these things called lipoproteins. These are particles, and they can actually interfere with the CBC analysis. So, and with the cell counts, for example. So therefore, one needs to be aware of some of these pre-analytical issues. But there are many others, as listed here, that can cause problems, including, fragments, giant platelets, RBC fragments, and so on. This table is, my suggestion is to go and, if you're interested, to look at the publication in the International Journal of Lab Hematology. It lists all of the sources of these errors. These are the more common ones. There could be others. But these errors have the potential to really compromise quality of lab testing. And in some cases, the problem might be there, but might be unknown to the lab staff. So results might be reported in error due to some of these concerns. So to prevent these errors, there are a number of steps that need to be taken, such as having standard operating protocols. So having clear standard operating protocols, providing training, extensive training to the lab staff, having sample rejection rules. So having appropriate steps to reject samples that are inappropriate, and also having quality indicators in place. So each lab really has to have a set of quality indicators to monitor quality. In this case, at the pre-analytical step or phase, but you could also have quality indicators for analytical phase and post-analytical phase. Some instruments have started using new automated technology to try to detect some of these errors and correct for them. An example is actually the Monday instrument that uses this interesting feature called optical platelet detection, which allows for correction of errors that could occur, especially with low samples with low platelet count. So this is shown, and it's actually, there is a couple of reports on this, one from Chile, there's also a publication from China that you could look at. But basically, this is a feature like platelet O that can actually correct for this kind of platelet problem. So it's an example where instruments can be actually helpful in detecting some of these potential pre-analytical issues that can actually assist in preventing these errors. And this can be a relatively common problem, and therefore this feature can be very, very useful. Another important pre-analytical consideration is utilization. I already mentioned that test selection and utilization is really important. And labs can work with clinical areas to ensure appropriate test selection and appropriate test utilization. Now, in Canada and the US, a program has been developed called Choosing Wisely. Now, this one is called Choosing Wisely Canada. There is also a Choosing Wisely US. So these programs have been trying to, started actually in 2016. The idea is to ensure appropriate test utilization. So prevent overutilization, but also prevent underutilization. There is evidence that in many cases, CBC testing, for example, is overutilized. But in some cases, it's actually underutilized. So the key is appropriate utilization to prevent both over and underutilization. Now, this is actually a publication that talks about choosing wisely recommendations and when to, for example, to do serial blood counts and when not to do testing that frequently. Also on thrombophilia and other areas of hematology testing. There are a number of recommendations. So I welcome you to review the publication and some of these recommendations because it can be very useful. And these are evidence-based recommendations to improve utilization. So this is also, again, a pre-analytical issue before you actually even do the testing. And utilization is becoming more important. Here at our hospital, we have a committee we have a committee on test utilization, and we are discussing between physicians and the lab how best to select and utilize testing, not just in hematology, but in chemistry, in genetics, in pathology, and other areas. Now let's move on to analytical consideration. And analytical considerations means when you actually are doing the testing on your instrument of choice, there are a number of key aspects. Of course, you have to ensure that the instrument is appropriately validated and verified. Normally, the validation is done by the manufacturer before the instrument is sold to the lab. But then the lab is responsible for verifying the instrument performance, and also continue to do internal quality control, external quality assessment, and also ensuring appropriate use of reference standards. This is particularly important in chemistry, less so in hematology, but these are steps that are important to ensure continuous high quality performance of the instrument. So here I say that it's important to have quality measures and indicators and have these established to monitor analytical quality. Now, the first, in terms of analytical quality assurance, the first step is internal QC or IQC. And this is, of course, commonly done by all labs. Manufacturers typically have recommendations on the frequency of IQC, normally at least once per day or once per shift, where you could use 2 or 3 levels of quality control to monitor the performance of the instrument. Now, in most cases, commercial QC is used, but in some cases, so-called homemade patient samples can be used to monitor the instrument. So it's an essential component of quality assessment in every lab. And typically there are 2 or 3 levels that you could use, low, medium, and high. And these typically detect analytical issues requiring either recalibration or the use of new QC materials if the QC material is faulty or expired. And in hematology, typically multi-parameter control is used because multiple parameters are being measured as part of CBC, and therefore you need a multi-parameter control. And the typical approach is to use a Levy-Jennings chart to monitor. And I'm sure you're all aware of this, and your labs are routinely doing this. But it's important to establish appropriate limits so that you know what to flag. Now, the different instruments, different manufacturers have different recommendations. Some suggest 1 SD as flag, some recommend 2 SD, but there are also published guidelines, and I will mention that. So in Canada, there is this institute called Institute for Quality Management in Healthcare. This is the accreditation body actually for labs in my province in Toronto, in Canada. And they actually did a survey of quality control practices in hematology. They surveyed over 180 labs in Ontario. Ontario is our province. It's about 15 million people, and they did a fairly extensive survey, and this was published. The results has been published back in 2018, and they found that, of course, all labs are doing quality control, but most of them are using commercial QC. Some are using patient samples as well. So they supplement commercial QC with additional QC material. And they typically follow manufacturer recommendations. So again, it's important what the manufacturer is recommending because that's mostly what labs follow. Most labs actually do the quality control at the beginning of the day, but there are different practices. So they found variation in the practice. And some of the key reported quality control practices were assessed. And these are some of the key observations that most labs followed precision goals from the manufacturers, or some actually use published precision goals. Some are using standard deviation of the control quality control results, and but they also compared that to what allowable performance limits are from IQMesh. So in Canada, there are recommendations and performance limits that have been recommended. And therefore, labs have different sort of approaches. Some are following these performance limits. Some are following, actually, most people are following manufacturer stated recommendations. Some more information from this survey. They found that all labs use 3 levels of QC, and 80% of the labs not only use commercial, but also use in-house patient QC, as I mentioned. All labs used established QC rules and limits, and as I mentioned, based on different recommendations and different goals. And I've already sort of mentioned these in the other table. So their conclusion was that there is a need for consensus practice recommendations and precision goals, essential need to guide and standardize QC practices in labs across Canada, well, truly across the world in every country. One area of that's been introduced in hematology and for a long time in chemistry and more recently in hematology, the concept of using sigma level. Now, sigma level is used by manufacturers like car manufacturers. Auto manufacturers use sigma to assess the level of quality in their production to minimize errors. So usually 3 sigma and above are acceptable, or those 6 sigma is ideal to achieve 6 sigma quality. So this is a quality metrics called 6 sigma metrics. It's a widely applied quality indicator, especially in chemistry, now increasingly used in hematology, and is based on, it's a composite a statistical measure based on the assessment of total allowable error, total imprecision, as well as bias, method bias. So some labs have begun actually in the hematology area to use 6 sigma, and this is one publication that looked at, back in 2016, that looked at the hematology parameters and sigma level. You notice that most parameters actually had sigma level above 3. So anything above 3 is acceptable, but some actually had sigma levels above 6. So that's quite good. That's so depending on the instrument, you may have different sigma level, and this is for level 1, this is for level 2. You can see performance was pretty good when you use sigma metrics to assess quality. Another key aspect of analytical quality is external quality assessment or assurance. And there are many, in the area of hematology, the main sources of EQA around the world are WHO and some companies such as Biorad, UK, NSQ, this is mostly in Western countries, in Europe, in North America, Randox. There have been a number of other, I'm sure there are many others. These are some examples of EQA. And I have to tell you that IFCC is starting to develop a program as well in quality management. And we're hoping to use EQA to support, especially developing countries in some of the, in Africa, in Asia, in Latin America, in other parts of the world, to assist some of the clinical labs. Actually, 10 countries have already been selected to start the program this fall. So it's called the IFCC Global Lab Quality Program. And we are actually working hard to start assisting countries with external EQA. This would be EQA initially in chemistry, but we're hoping to also include hematology in the future. Now, so usually EQA is an important part of quality assurance, and it tends to be important, especially when you're seeking accreditation to ISO standards 15189. And usually a number of parameters are covered by EQA, but this varies from provider to provider. So clearly, if possible, EQA should be done. Our hospital has frequent EQA surveys that assess, these are like blind controls that assess the quality of the labs externally. Of course, you have internal quality assurance, but external quality assurance is equally important. There are a number of concerns, especially for hematology, that you should be aware of with EQA challenges. The truth is that there is no ideal material. Samples tend to be unstable, and so commutability is a concern. So stability of the sample is an issue. Blood cells are inherently unstable. Therefore, in order to make EQA samples feasible, usually they are treated with to improve stability. But that treatment can cause changes to cell morphology and therefore can actually affect results. So this should be kept in mind, that EQA samples are not the same as patient samples. And methods of stabilization, as I mentioned, are important because they can affect morphology, but also can affect results. So additional considerations, this is actually a survey that was done relatively recently in 2018. We're looking at 142 labs actually in China, in Beijing, and it was a 2-year study looking at 6 PT. PT stands for proficiency testing, which is the same as EQA, or external quality assurance, or sometimes called proficiency testing. This was done, and they found that 90% of labs actually were able to perform well and fulfilled CLIA analytical code. CLIA is a US-based sort of organization that monitors quality of labs, and the analytical goals were met or fulfilled based on this survey. There was less concordance for some of the other markers like hematocrit, MCV, and so on. And clear analytical goals for coagulation tests, however, were also pretty good. We're satisfied by nearly 90% of the labs in Beijing. So this was actually good news in the sense that most labs appear to be able to meet the EQA performance specifications. Standardization is another concern with hematology particularly. It's been a little easier with chemistry assays to standardize, and actually IFCC, the International Federation of Clinical Chemistry and Lab Medicine, has been involved for almost 70 years. We actually are celebrating our anniversary next year, a 70th year. For 70 years, been involved with standardization, but the standardization mostly focused on chemistry. The reason is that in hematology, standardization is even more difficult because, for example, CBC is a multi-parameter analysis, and in having appropriate standards is very difficult, particularly since you're talking about cell analysis. And analyzers use different measuring principles, such as optical methods, impedance methods, fluorescent methods, to detect cellular characteristics. Therefore, standardization is a clear concern. This has been discussed in a publication that I've listed here, talking about how one could approach preparing standards for improving a standardization of hematology testing. So I invite you to look at that paper. I should go faster because we're running out of time. Analytical considerations, when you look at methods, actually it's interesting that when this study compared 5 different analyzers, CBC analyzers, and hematology instruments. And they found that for some assays like WBC count and RBC count, there was very good concordance. But, and this is another for hemoglobin, hematocrit. But there were other assays like MCV or platelet count where there was major differences. So for the nice thing, and actually in our own hands, we have studied a few different analyzers, and we find very good concordance in measurement across different instruments. So that's good news. But there are some analytes like MCV, platelet, where the measurement is not as consistent. So I should go faster. Finally, post-analytical considerations, which means reference intervals. You need to have appropriate reference intervals to interpret results. And so establishing these is important, or verifying them. So there are many groups around the world where they have looked at reference intervals, and especially in chemistry, but also in hematology more recently. I have mentioned some of the studies. Caliper is our program in Canada. There is Happy Kids in Australia, KICS study in Germany, Child X study in the US, and there are others. But actually the Caliper program in Canada has turned out to be the most active, most largest program. And most of the world actually in US, Canada, Europe, Asia, Latin America are using the Caliper data. So Caliper has become sort of the main source of reference intervals, particularly for children. And so this program, those who are interested can go to our website. It's caliperproject.org. And the database is called caliperdatabase.org. So those websites can give you a lot more information because I don't have a lot of time. But this program has studied over 12,000 children, and we have established reference intervals for over 185 tests in children. That includes hematology recently, a few publications on hematology. So those are all available. You can actually search Google for Caliper Project, and you can get all of the information readily. So these are some of the studies we have done on hematology. Studies have recently been completed and have just been published. We have recruited many children from 1 year of age to 18 years of age and tested them on multiple platforms. This is some of the data. So I recommend you look at our publications recently just came out last year, American Journal of Clinical Pathology, 2 papers, as well as International Journal of Lab Hematology. So these 3 papers actually explain some of the data that we have on hematology markers from birth to 18 years of age. So we have established reference intervals, and we have graphs as well as tables of reference intervals for all of these markers. This is, I'm going to, for sake of time, pass this and just tell you that we have just another paper just accepted for publication. This is doing linear reference ranges, not just tables of reference ranges, but doing continuous linear curve calculations. So this is actually probably better for hematology markers like RBC, hemoglobin, and others to have continuous reference ranges rather than partitioned reference ranges. It's a new concept, but it's a very important, perhaps, tool to help labs and to help clinicians. Finally, I wanted to let you know that we are collaborating with Monterey to start a study. The instrument has actually been installed in our lab, and we are going to do a full study of reference intervals in children on the Monterey instrument. We are using the BC 6800 plus analyzer, and we'll be establishing age and sex, but also ethnic specific reference intervals, comparing Caucasians and Latin American children and other ethnical groups as much as possible here in the Toronto regions, because we have many people from different parts of the world. We will be publishing these results, so we'll also be putting them on the Caliper database. Hopefully by later this year, we will have the results to publish. So I just wanted to let you know that we have an app as well, a Caliper app that you can actually freely download on your mobile device, either on iPhone or on other phones. The Google Play also has the app for download, as well as iTunes. So they are available for free. And this allows you direct access to the database for all over 180 different tests that I mentioned. So the online database I already mentioned as well. So I want to end here by just summarizing that quality assurance in hematological laboratories is essential, and you need to be aware of issues, considerations for pre-analytical phase, analytical phase, and post-analytical phase, and basically controlling quality of the total testing process in the lab is really critical. So thank you very much.
Thank you so much, Dr. Adeli. Such a fruitful presentation. I've learned the importance of quality assurance for the whole process of analysis, including the pre-analytical, analytical, and post-analytical process. It is surprising that just a moment, let me share the screen first. It is surprising that 70% of the errors comes from the pre-analytical process. And also, a very good analyzer would help you to minimize the analytical errors. Don't forget to do a QC at least once a day, right? If it is possible to do to join an EQAP program to ensure the quality the result accuracy. So, thank you so much for sharing the Monterey study on the Trojan intervals reference setup. It is very useful for us. Now, let's get into the Q&A section. If you have any questions to ask about the quality management in hematology or others, please feel free to raise your hands in Zoom or leave your comments in the chat box. I've seen several questions already in the chat box, so we will connect your voice in the meeting room or forward your questions to Dr. Adeli in a moment. Today, we are honored to have another expert, Dr. Henry Wong, in the room. Dr. Henry Wong is the Senior Director of Medical and Scientific Division of Monterey IVD, and he is very experienced in oncology. So Dr. Huang, do you have any questions to ask Dr. Adeli first? Hello, Dr. Huang.
Hello, Dr. Wang. Dr. Wang.
Hello, haven't seen you for ages. How are you?
袁女士觉得我们这边就是我们医学所可能就是
can you hear
在
me?
这方面是有一定基础的，是可以去做的，是吗？对，就这方面不是如果我
Hi,
们能
Dr.
够
Huang.
我们
Hi, Dr. Adeli.
How are you?
Good.
Andy?
I am having another meeting now.
I know you have another
Yeah,
meeting.
meeting
So
conflict.
I can. Don't worry. I don't want to bother you because you have another meeting, but I can try to answer the question.
Yeah. Actually, I have gone over your precision briefly. So I think it's very interesting, and then I also have known that you have done this a lot. So you are very experienced on in this field. So and
thank
also
you.
yeah, and also I have visited your laboratory, your university in Toronto. So what I have seen is amazing.
Thank you.
Yeah, think about the quality control of hematology parameters. So we have launched a joint study for the parameter in children in different races, right? Different ethnic. I would like to know if you when you collect the sample, so how would you control the variation between sample and different from different location, from different group of kids? So I would like to know the procedure in control the quality. So this is my concern.
Yeah, certainly.
Okay.
Yeah, I can answer that. So we have been collecting samples in different schools.
Yes.
Like we go to the school and we collect samples. We also have collected samples in the hospital from the healthy siblings of children who come to the hospital. So when we compare, the differences are very minor. And so it suggests that the collection procedures are fairly standard and consistent, because I can show you some of the graphs. You can see there is a lot of good consistency. Variation seems to be relatively low when you collect, of course, appropriate conditions, under appropriate conditions, and also you analyze, as I mentioned, within a few hours. The variation tends to be quite low.
那个秦江江老师，秦老师您在吗？因为我知道我们医院其实有很多临床的专家都有跟医生做一些临床的交流和沟通。我不知道你们在这样的沟通中。
For the parameters of hematology in children, so we are going to have more than 60 parameters, right?
Yeah, quite a few parameters. I haven't counted the total number, but it's quite a few, yes.
Okay, yeah. So I hope that when the project is done, so we are going to publish some kind of papers or consensus, and then it's going to be our, I would say, a big job in overseas, so we can we can educate our customer that we have done this work with you, with a famous professor in the world as head of IFCC. So it would help us to educate our customer and then help us our product marketing, right?
Yeah, certainly. No, we always publish all the data. Of course, we're going to see at least one publication, maybe more than one publication, depending on the data. For example, we could have the age and sex stratified reference intervals published in one paper, then ethnicity, effect of ethnicity, another paper. So there could be, definitely we will publish the data, and then we will make it available to all of the customers so that they can use the information. Actually, this is how we have worked for the last 10 years. That's why all of the data we are using right now are being used by hospitals around the world. So, for example, the data we have on Beckman and Sysmex are already being used in Europe, already being used in North America. So, so we will do the same with the Monterey study, of course.
Yeah, good. Thank you very much. And how have you been during the pandemic in Toronto so far?
So far, so good here. I'm fully vaccinated, fortunately.
Oh, okay.
Canada vaccination going well. So I had 2 doses of the vaccine. So I'm actually traveling this Saturday. I go to Dubai, and then I go to Italy. So traveling is starting.
Okay. Was the vaccine from Pfizer?
Pfizer vaccine,
okay.
yes.
mRNA vaccine, okay.
mRNA.
Okay, okay. Yeah, I'm going to visit my family next month, July
very
15th.
Good.
Yeah.
Yeah,
but
that's
I may
cool.
not be able to go to Toronto.
You think you can or no?
No, I don't think I can.
Yeah, right now the problem is that the border is closed, but it might be open in July. It might be open.
Okay. Let's see. Let's see. Okay.
Okay. Thank you so much for Dr. Huang and Dr. Adeli. I've seen Dr. Ramon raise up your hand. Hi, Dr. Ramon.
Yes, yes.
Hi, how are you?
Hello.
Do you have some?
Hi, Dr. Ramon.
Adeli?
Yes.
How are you?
I have a question for you, Dr. Khosrow.
Please.
Looking the reality of many labs in the world, many labs receive samples that are transported for 24 hours, 30 hours, even sometimes 40 hours after the blood draw. There is a quality control for sure in the lab, that they have the quality control materials in the fridge, and they analyze that. But which initiatives we have today to control the transportation of the samples, like to include with the racks you have with all the samples collected in a peripheral lab, and to transport the control together with the samples to see how the temperature, sometimes in summer or whatever, how that affects the quality of the samples.
Very good question, Dr. Ramon. Of course, the recommendation is less than 8 hours. It should be kept at, it could be kept at room temperature for up to 8 hours. Ideally less than 4 hours, but less than 8 hours is acceptable. But if it's going to be more than 8 hours, the sample should be put in a fridge or transported on ice. It's not ideal because it could still impact some of the analysis, but definitely if a sample is going to take 24 hours before it arrives in the lab, it should be kept on ice. It should not be kept at room temperature. This is a challenge, of course, and it could lead to significant, actually, pre-analytical errors, as I mentioned, where you will have significant changes in cell morphology. Some of the cells might be degraded and destroyed during the transport, and it could affect the results, of course, of the CBC results. So therefore, transportation, if it has to be overnight or more than 8 hours, then it needs to be refrigerated. Still, there is a risk, but it's better than room temperature.
Yeah, I have been, because I am part of the International Council for Standardization in Hematology, we have been evaluated to transport hematological samples is better at 4 degrees, even if we know that affects some parameters, but is the best. But my question, of course, when you have a big lab and you receive samples for many labs, you control, you establish certain standards, okay? That the samples have to be transported in this way, blah, blah. But then my question is quality control, because you set up something, but samples can be, sometimes the mailing of these samples can be wrong, can be delayed. And I miss initiatives to include vials of quality control together with the samples when you transport samples from one lab to another lab. And that is what I was suggesting.
I see. That is a very good concept, actually. Yes, a very good suggestion that one could potentially include quality control samples as a sort of a indicator, as a quality indicator during transport. It's a good suggestion. You could, but the quality control samples have to, of course, have to be stable as well. So depending on the choice of the quality control material, it may or may not be as stable. So, but it is a good suggestion, but I don't think it's done routinely. Unfortunately, it's not done.
As far as I know, it's not yet established, but I propose that to be included in the future.
It's a good proposal. I like that idea. It's a very good idea.
Good.
Thank you.
And then my second question is about reference values. In hematology, we are continuously developing reference values, and one of the last initiatives was to establish your own passport. For example, even if you have an hemoglobin of 12, but if your hemoglobin has been 14 for 3-4 years, and one day falls to 12.5, this is abnormal. For the reason to have like your personal reference values, and today,
exactly,
because the computers are every day, to have like your passport and to consider abnormal everything that goes out of two standard deviations of your regular values. What do you think about that?
Yes, there was actually a recent publication on this called Personal Reference Interval and the concept. It's been promoted. Right now, it's not a routine practice, unfortunately, because you know, most patients are not tested that often. So so unless you have a situation where a patient is tested, let's say every 6 months on a routine basis for many years, then you have a very consistent data set that you can use to establish a personal reference level. And then as if that patient develops a disease, that level can change and you can detect that change. Because this data is very limited, maybe only a few patients have that kind of information. Therefore, all labs right now use population reference intervals based on a population, a sort of apparently healthy population. This is based on the recommendation of the CLSI, as you know, the Clinical Laboratory Standard Institute, or CLSI. So the recommendation is to use population reference intervals. Right now, I think the personal reference interval concept is good, but it's not feasible at this time.
No, and I think it's not feasible also because many laboratory informatization systems don't permit that, don't permit to see if once a patient with chronic lymphocytic leukemia that will have it maybe for a long time, or beta thalassemia, that once you make a diagnosis, the diagnosis is kept and you don't need to do it anymore. And again, also control that you were here 4 years ago, you get this hemoglobin, and now you get a new hemoglobin, to permit to have a historical data of the patients. And that is also a challenge for the companies, I think.
Yes, I think actually, Dr. Ramon, best to use both. My idea is to have the population reference interval, particularly the continuous curve that I showed. Then you can plot your patient result and then monitor that patient reference over time based on the population data. This way you can assess whether that patient's result is changing over time.
I think this will be even great. Great idea. Yes.
It would be very helpful.
Yes.
Yes.
Thank you, Dr. Raymond and Dr. Adeli. I have seen a question from the chat room from King Khan. His question is, for sigma calculation in CBC, is there the best recommendation for percentage bias calculation from EQA or from mean peer instrument are suitable for the best recommendation? Thank you.
Yes, there are guidelines available as to sort of total allowable error for different parameters like hemoglobin and hematocrit and MCV and others. So one needs to look at those recommendations. In Canada, we have recommended allowable error from our IQMH, the Institute for Quality Management in Healthcare. But there are also clear recommendations in the US. There are also other European recommendations and others. There might be recommendations in China as well. So therefore, you need to go based on those recommended levels before you calculate your sigma level. So the sigma calculation should be based on guidelines, based on recommended allowable error calculations.
Okay. For the next questions from Coco, she asked, how do lab doctors play a role in multidisciplinary, I don't know how to pronounce this word, multidisciplinary, oh my God,
Multidisciplinary,
yes, team in Canada. So do you know how to answer this?
That means that you need to participate regularly, participate in their discussions, they have rounds all the time, patient rounds, and they discuss patients. So the lab scientist or the lab physician participates in those discussions. And once they get to know you, they will include you in their clinical decision-making. They will include you. They ask you, they consult you for what tests to select and how to interpret test results. And if they have problems, they consult you and ask you to help them. So we do that here in our hospital. We regularly meet with the physicians. For example, with the endocrinologist, we regularly meet and they ask us questions and we answer. We also have a regular email communication. So therefore, we are sort of part of the team, and that's the key. So the multidisciplinary team at the center of healthcare, that means that you're part of it. You're not, the lab should not just be a service. It should be part of the team to provide care.
Okay, thank you, Dr. Adeli. Let me share the screen. Can you see my screen right now? There is some questions from the LabClub. Can you see?
Okay.
The first question is from Mohammed Lutman. How to quality check of an instrument? This is the first question from him.
Yes. So actually, it's a good question. So when you buy a new instrument, or if you have a problem with an instrument you're trying to troubleshoot, usually you go through a verification protocol. It's called an instrument verification. So the instrument verification usually includes looking at precision, so looking at reproducibility. So you run quality control material to determine your CV or coefficient of variation. So that's called a precision check. Then you also need an accuracy check. You do a comparison, a method comparison with another lab, with another instrument. So ideally you use patient samples to do a comparison with another lab. A third parameter is linearity. You could use linearity samples to look at how linear the results are. So it's called linearity check. You also should look at interferences and carry over some of these other parameters, sort of parameters that you can assess. But most important and most common is the precision check and the accuracy check. So quality control, running quality control usually several times per day over 5 days, then you can get an idea of how consistent and how reproducible the result is. And the second is method comparison with another instrument, with another lab. There are other checks you can do, which is good, linearity, carryover, interferences, but those first two are more important. So that's how you assess quality of an instrument.
Oh, very comprehensive explanation for verification tests. Okay, for the second question, what is the criteria for critical result as well as how to report a critical result? So it's like a
So actually, it's a good question because there are actually critical values or critical results for a specific hematology test. So you should look at guidelines. There are published guidelines that define critical values for different tests. For example, for platelets, there is a low threshold critical value, and there is a high critical value. So too high should be reported as critical, and too low should be recorded as critical. Different levels. There is also a critical value for hemoglobin and other parameters. So yes, there are guidelines, published guidelines. If you email me, I'm happy to share with you some of the publications and guidelines around critical values.
Okay, I think the third question from Godwin is similar to the Muhammad one, right? How are you going to verify a test result and action to take when a result is seriously abnormal and unexpected? I think you have answered it already, right? Do you have any more?
Actually, I could add a bit more to that, and that when you have a problem, so when a sample is very abnormal or has an unexpected result, you need to first check your instrument to see if the quality control is acceptable, if the instrument has acceptable performance. If it does, then it's not the instrument, it might be the sample. So there are two the steps you take. You repeat the test results, so you repeat the testing on the same sample, and if the result is still abnormal, then you ask for a new sample from the patient in case there is pre-analytical error. So if there is a sample problem. So therefore, yes, there is this is called troubleshooting, where you troubleshoot and you try to determine the cause of the abnormal, whether it's a true result or a false result. So those are the steps you take.
Okay, thank you, Dr. Adeli. I've seen a last question. Let me share the screen first. You see my screen. From Betty, right, is here. Can you see it? Question or,
let me see.
Or chat box? Can I share? Oh, okay, it's here. Okay, the stabilize of the CBC control, for example, MCV, can be changed up to FL from the first day or used till the last day. In this case, we will have an imprecision. When we calculate sigma, it's quite not good. How we can solve the problem of the parameter that the control material is not stabilized?
Well, I think, yeah, if the material is not stable and you're not, especially for MCV, and you're getting variable results, it's very difficult to calculate a reliable sigma metric. So that's why if you look at the publications, they say that the sigma metrics work well for some parameters like hemoglobin, hematocrit, and white blood cell, red blood cell. But for MCV, in the publication, it didn't work well because of this kind of variability, because of instability. So yes, it is a concern, and it's difficult to calculate a sigma metrics for MCV until those the measurement is more and the sample is more stable.
Okay, thank you. Hi, Dr. Ramon. Hello. Do you have
Yes,
any
I
more
would like to
questions?
share the ICSH critical values, if you don't mind, Dr. Adeli.
Please.
That we recently published that, and let me, if you permit me to share my screen.
Sure.
Sure.
I will stop sharing and let you share the screen for us.
Wait a second.
Sure.
That I can see that, yes. Let me see. Where it is that? Wait a second.
Sure.
Here. We did recently a review of the critical values in hematology, and the ICSH published this test. In the same article is all the method how to report it and how to deliver, and here is a review of many previous articles and many previous proposals of critical values. The one I was using before that was the Emancipator in 1997. And I want to share that everybody is a free download in the internet, and everybody can have it. And of course, the CLSI have published also its own, but this is what I want to, everybody can access to these critical values from the International Council for Standardization in Hematology.
Thank you, Dr. Ramon. Yeah, this is another very good sort of guideline document that I mentioned. There are a few different guidelines. So these provide the sort of cutoffs or critical limits that you can use in reporting your results.
Okay, that's all what I want to share.
Thank you.
Okay, thank you, Dr. Adeli, once again for answering all the questions, and thank you, Dr. Ramon, for the supplementary information. It is all very useful for us, and we have lots of information today. So happy. So thank you so much.
Thank you, Shan, and thank you, Monre, for hosting this presentation and QA session. Thank you, Dr. Ramon, for your great participation. If there are any questions, feel free to email me.
Okay, thank you.